Alright everybody, this is the 2017 paper 2. Remember, this is 2 hours and 15 minutes. You can use a calculator on the exam, so make sure you have one. There should be some in the testing room. So remember, start off by reading and underlining everything. Here we were looking at this Chinese soft-shelled turtle living in saltwater marshes. They go underwater, out of water. It's talking about, so they have regular lungs, regular kidneys, but they have these special little microvilli in their mouth. So there was this hypothesis that the turtles could excrete nitrogenous waste out of their mouth. So they designed a study, and this is an actual study we see in 2012 that AP biology doesn't have cool stuff like this. So that's their hypothesis. They can bring in oxygen to those microvilli and also excrete urea. Pretty cool. I think if we could do that. Excrete pee out our mouth if we go swimming. So they collected the excretion data from, remember IB always uses a scientific name. Turtle urinates both in water and out of water. When in water, it allows waste products to be washed out of its mouth. And I want to underline that. And then out of water, it regularly dips its head into water to wash out its mouth. So when it's on the shore, it'll wash out its mouth. So the tables show the mean rates of ammonia and urea excretion. Remember, ammonia is more toxic than urea. Ammonia, NH3, is converted to urea pretty quickly in your bloodstream and the bloodstream of animals. So here we have, just familiarize yourself with the data table. Excretion of the nitrogen, and we have ammonia and urea. When it's submerged in water, how much ammonia and urea is being released, and then when it's out of water, and this is in the mouth, right? So I want to highlight that. Use your highlighters, whatever you got, whatever you feel best with. And over here we have the kidney. Okay, in water, out of water. So the first question asks you deduce, which basically is IB speak to figure out. So there is one answer. Okay. If it asks you to evaluate, evaluate, it could be more than one answer. Meaning there's data for both sides. So when you're doing your IB test and you see evaluate, it's not going to be one answer or another. But deduce means there is one answer. So which one changes more when a turtle emerges from water? And the answer is urea, because we see ammonia 0.29 to 0.30, not too much. And then that's from the mouth. And then from the kidney, goes from 0.63 to 0.54. So it goes down a little bit, not too much. But we see for the urea, it changes pretty drastically when it's emerging. It's out of water, 0.9 to 1.56 and 0.07 to 0.73. And it would be a good idea to write the units per day per gram. All right. So that's just reading the table. Usually that'll be the first thing they'll do. Now we go to this other question here. compare and contrast the changes in urea excretion by the mouth with the changes in the urea excretion in the kidney. So you're comparing to the mouth and the kidney for urea. So remember, we have two sets of data, ammonia and urea. So we're just going to be focusing on this one for question number 2. So what we see is for both of these, it's kind of funny. They both increase by 0.66 micromoles, right? 0.9 to 1.56 and 0.07 to 0.73 is both a difference of 0.66. Further, you can do, the new minus old divided by old. All right, so taking that new number minus the old number divided by the old number, give you that percent difference. And you could do that in this question. And if you just look at that, in the mouth, it's already pretty high, and it goes higher. In the kidney, it's really low, the urea, when it's in the water. Maybe because it's relying on the urea being excreted by the mouth. But then it really jumps pretty high. So if you look at the percent difference, that kidney, it's a 940% increase versus a 73% increase. Like, dang! If you're looking at increasing things, that's a much more significant. So that could be, a contrast. They both go up, but much higher in the kidney. And another thing you can say, more excretion of urea by the mouth in the water and out of water. So it looks like you just have a lot more urea coming out of the mouth, whether or not it's in the water, whether the turtle's in the water or the out of water, or if it's out of water. So that's just pretty interesting, development or piece of information there. So this, usually IB, the first 4 pages are going to be all on one question, and they just kind of feed off the other and kind of tells a story. Storytelling. So talking about the behavior of the turtles rhythmically moving their mouth to take in water from the shallow source, then discharge it. And then they decide to look at the dissolved oxygen and the quantity of accumulated urea in the rinse water. So this chart right here is the urea, and over on the y-axis here, we see the percentage of the oxygen in the water. So here we have, 4 micromoles, and over here we got 20, we got 38. So we can see in this graph, the urea increases in water over time. So we can see that urea coming out of the body of the turtle into the water. And we can also see a general pattern of a decrease in the dissolved oxygen. So describe the trends in the graph for dissolved oxygen. So we can see it decreases when the head is dipping. So this triangle is showing the dipping, and we see decrease here, decrease here, decrease here when you see the dipping. And then we see it kind of rising. You see right here that dissolved oxygen in the water goes back up when that head is not in the water, because it's not sucking that water out. But here's reasons for the trends. It's being absorbed from the water when dipped into the turtle, so that water, or that O2, is going into the turtle. And that oxygen is being used for aerobic respiration for the turtle. When the head is out of water, like here, water is dissolving into the water. So basically diffusing. from the air into the water. So that's what happens normally. You put any body of water, out there, oxygen from the air is going to diffuse into the water. You can speed that up by, say, mixing it up, disturbing the water surface. So here we go. We're going to go look at a urea transporter. Remember, this is a type of protein. It's a protein in the membrane that helps to move. We have glucose transporters, water transporters, and of course that sodium potassium pump that we went over before, to create that negative charge in your neurons. So there is a known inhibitor called fluoretin. Fluoretin, known inhibitor of membrane proteins that transport urea, was added to the water, in which further set of turtles emerged their heads. So these are new turtles, it looks like. Maybe we need to get some new turtles. So they're adding this inhibitor that prevents the urea transporter from working. And I want you to do this too. When you do your exam, I want you to write all over it just to kind of clarify things, because I need to when I go over these. So here we have the rate of excretion. This is the control, without the fluoretin, and this is with the fluoretin. Check this out. Almost no urea was excreted. So it looks like there's definitely this urea transporter protein. So deduce with the reason whether the urea transporter is present. So deduce, remember, so there is one answer. Okay, it's not evaluate. Remember, the other main question they might ask you, or term they might use, is evaluate, which is 1 plus possible answers. So many evidence for 3 different ideas. Definitely have the result that urea transporter is present because that fluoretin, if it wasn't present, can't really explain this result, could you? Seems to have just completely shuts down that urea transport. So almost no urea transport when you have fluoretin there. All right, and here we have some gel electrophoresis. So we're looking for messenger RNA expression of the urea transporter gene. So it took different tissue samples and they looked for this mRNA activity. Remember, for the gene for the urea transporter. I wish they gave you the name, because I just have to kind of say that. Maybe it's really long. But they looked at all these different tissues. And what do we see? For the tissues that are in the tongue and the mouth, we see evidence. So this means that the RNA is present. Or the mRNA. It shows up as these bands for, we'll say, RNA fragments, right? You know that gel electrophoresis creates these fragments and put them through a thick gel, and it will get stuck in certain places. So the gel shows that there is mRNA activity for urea transporter gene, the urea transport gene in the tongue and the roof of the mouth, but not in any other tissues, because they don't need that, right? Your body's only going to transcribe things that are being used. So we see, since those are being used in the tongue and the roof of the mouth, we see evidence, those bands. And we see how the band is stronger in the tongue. So we can assume that more urea is excreted from the tongue. We should put that too. More urea is probably the tongue, since it has more mRNA activity for the, if I knew the name I could just write that, but for the urea transporter gene. Okay, so that is question 1E. And here we go with another question about our turtles. So the expression in the mouth was assessed using that mRNA activity. They were kept out of the water for 24 hours, then injected with either a salt solution that was matched with its normal tissue, dissolved ammonia, or urea, followed by another 24 hours out of the water. And we have the mRNA expression, okay, of the urea transport. So this is the turtles. You know, they were injected with the salt solution, so that's our control. And here we have the expression when they were injected with the ammonia or urea. Hopefully they did proper, animal, welfare controls here. So which of these turtle groups represent the control, giving a reason? So that would be the salt solution, because it doesn't have any extra nitrogenous waste products. It matched the salt concentration of the turtle. So you might ask, why inject it at all? Well, anytime you do an experiment, you inject something, you do a treatment, that treatment itself could have an effect on the outcome. So you want to do the treatment in all your groups, and usually it'll be a placebo or something. Remember, because if you get like a pill, you might start feeling better, because psychologically you think, oh, I'm getting treated now. And your mind has a lot of really complex effects on how you feel and how disease works, to an extent. You still can't think yourself out of cancer, okay? Things like that. Some people, you will see ads for people, con artists will try to pull that one on people as far as medical things. Like, it's just in your mind, and you take this drink, green juice, and it'll fix everything. Okay, so we know that's our control. Let's move on to here. Suggest a reason for the greater expression when we see greater expression when we see it dissolved in ammonia. So one reason is ammonia is really toxic, so you really got to remove that quickly. The other thing is ammonia is converted to urea. Okay, and the difference really might not be significant. You see how there's overlap here? So this top of the bar overlaps here. So might not be the difference. Obviously means overlap means it might not be statistically significant. That means the difference we see between the two groups. Now it depends. The statistical significance, you would have to know what those error bars mean. If those are the confidence intervals, if that's the 95% confidence, then you definitely be not significant. But If it's something like usually in IB we're using standard deviation, it may or may not be significant. But that overlap, because we're just seeing this is, you know, the general area. If you did it again, sometimes that experiment, this level might be higher than here, if this is the kind of range we're looking at. So it might not be a real significant difference if you see that overlap. Okay. And then we have poor turtles. Sometimes they dry up to small pools. What problem could that cause to nitrogen excretion, and how their behavior might overcome that problem? So as those pools dry up, there's going to be less of a gradient, because the urea is going to be more concentrated in the pools. And if you have a less of a difference in concentration, you're not going to have as much diffusion. So that could be a problem. Like if you're dipping your head into water that's already pretty concentrated with urea, you're just going to have less urea coming out of your body. Less water is available to create urine, you know, and therefore, you know, have a way to, you know, get rid of the urea, because you need, you know, when animals moved onto land, you know, when you go out of the water, you're automatically going to have to worry about drying up. So, you know, we have to have this balance between, you know, preserving our water and then releasing it so we can get rid of our waste products. So that's why your body is always adjusting the concentration of your urine, because it's got to conserve water on one hand, it's got to get rid of urea on the other. More expression of the Urea Transporter gene to speed transport of urea out of the cell. So yeah, so the turtle, its cellular mechanisms might ramp up the production. You might see, so if you did this experiment when the turtle was in those small ponds, you might see even bigger bands. Like maybe the roof of the mouth might be a lot darker, or a lot more evident, because now it's having to ramp up the amount of transporter proteins in its cells so it can get rid of more waste. And it might have to migrate to pools with more water if it can. You know, if there's pools nearby, you know, animals can move. And it might do more head dipping again, which is going to create you know, more urea. It's going to dip longer, and that's going to take more energy from the turtle. So those are all some things the turtle might do to overcome this issue. Alright, so that's done with the turtles. Let me break, and I'll do another video.